Compartmentalized microfluidic devices allow neurons to be studied in their highly polarized form. These devices are instrumental for both basic and translational neuroscience research. This protocol describes how to use cyclic olefin copolymer chips to compartmentalize neurons differentiated from human stem cells. These COC chips produce healthier cultures of differentiated neurons over PDMS multi-compartment devices.
In this protocol, we demonstrate the culture of neurons differentiated from the NIH-approved H9 stem cell line. Similar procedures can be used to differentiate neurons from iPSCs.
The multi-compartment chips must be placed into secondary containment with extra humidification. The channels of the multi-compartment chips must be filled with a pre-coat solution and then flushed with PBS. To begin, dissolve poly-L-ornithine in cell culture-grade distilled water to make 600 microliters of solution per chip. Aspirate the remaining PBS from the wells, making sure that the pipette tip is away from the channel opening. Load 150 microliters of poly-L-ornithine working solution in the upper right well. Wait for 90 seconds and add 150 microliters of the solution to the lower right well. Wait 5 minutes and repeat the loading process with the solution for the left wells. Then, place the humidifier tray with chip at 4 degrees Celsius overnight. If using a petri dish, wrap it with parafilm and place it at 4 degrees Celsius overnight. Next, aspirate the solution from the wells, ensuring